What I've done here is I've taken my two homologous chromosomes and I made their sister chromatids. I made a tetrad. And then, remember how we separate the pairs of sister chromatids from each other and divide the cell? I'd end up with a cell that looks like this. I don't know why I colored them in. I use things. Make sense? You take your two pairs of sister chromatids, you separate them and you divide the cell, and then you line them up again and separate each one of these out into the 4 haploid cells. And so when you do that, you'll end up with cells that look like this. Big P, big P, big P, and big P. So what that means is that when a homozygous purple pea plant goes through meiosis, it makes haploid gametes that all have big P alleles in them. And if you follow the white homozygous, well, I'll do the same thing. I'm going to pull apart the pairs of sister chromatids and divide the cell. And then I'll divide them again, and we should end up with so if a homozygous white mates with a homozygous purple, what do you end up with?
Crossover of the peas.
So if a homozygous purple mates with a homozygous white, that means homozygous purple goes through meiosis and makes these gametes. Homozygous white goes through meiosis and makes these gametes. So when they mate, you'll randomly pluck one gamete from the purple and one gamete from the white. And so what do you end up with when you randomly combine any two gametes from these pools? You should end up with, and I hope everybody can see this. I'll draw one out here so you can. So if I take, let's say, this cell, and I combine it with this cell, what's the genotype of that individual? Heterozygous. So they're going to be big P, little p, and they will be heterozygous. What if I pick another random one from this parent and another random one from this parent? Will it ever be different?
No.
It can't be, right? It's always going to be exactly the same. You're always going to end up with big P, little p, a heterozygous. So what's their phenotype? Purple. So this is their genotype. Their phenotype So big P, little p with a purple phenotype. So this is a pain in the butt to draw out, right? Wouldn't it suck if every time you wanted to figure out a cross, you had to draw out meiosis for them? It would be a pain, right? So we have a special tool that allows you to avoid that pain. And that special tool is called Punnet square. And so in the Punnet square, myself enough space here. In the Punnet square, this is what you do. You first draw a square.
Seems reasonable.
It's a good start. Pretty good square. Then you simply, next to the left side of the square and on top of the square, you write out the genotype of the first parent. And then on the left side, you write the genotype of the second parent. So if I'm crossing together, let's say, how do I do it for you guys? If I'm crossing together a homozygous purple, I write their genotype there. And I'm crossing them to a homozygous white. I write their genotype there. Next, I take each parent and I split their genotype into the two letters. So I take big P, and I split it into those two letters, and I write them separately along the side of the square. The two big P's split. And I think I out of order on the thing up there. Draw a line between them. And then I take the genotype of the second parent, the white flowers, and I split their two letters. Draw a line between them. And then all you have to do to solve it is in each square, you combine the letters from each parent. So you say, purple parent gives big P, white parent gives little p. Purple parent gives big P, white parent gives little p. What's that? Okay. Purple P gives the big P, white gives little, and so on. So no matter what you do, you always end up with a big P, little p.